MSc Mushroom Biotechnology elective course. Major part you have completed today is rare opportunity in African countries where these Termitophilic agaricales are found. We are working on this genus for last 33 years. 33 years. This person. 33 years. These 32 monsoons I have collected and studied these species. Now, how to study Termitophilic agaricales? They cannot be studied only in lab, because somebody gives you a specimen like this, will get totally incomplete information. To study any species, small or big, plant or fungus, you need to know its all life cycle stages. You cannot just study one stage. From seed to seed. You understand? Not only vegetative stage, not only flowering stage. From vegetative, flowering, full life cycle. Today you have seen almost full life cycle. You have tried to retrieve the pseudorhiza in the field. Few minutes back we were out of this Goa University Science Faculty block. Today is 17th of July 2019. We are taking afternoon class here in the Botany Department Laboratory of Goa University in the Science Faculty block A. And we have 19 students, I believe, today for this practical session here, which is the only session on these species on Earth. No university in the world teaches termitomycology. I teach it because we are in the habitat of those species. Our laboratory is within walking distance from the species. This is rare, even in African countries, Kenya, Tanzania, Zambia, Zaire, Central African Republic, Congo, Republic of South Africa. There are universities, but you have to walk half an hour to go to the habitat of the Termitophilic agarics and then study, which is very tiring, exhausting, and by the time you collect specimen and all, there are lots of problems. So the best scientific studies are possible when you combine ecological, biological, morphological, taxonomic and diversity information along with the processing of the different types of taxonomically interesting tissues, and complete the micromorphological, microscopic and microchemical observations on vegetative tissue, reproductive tissue and reproductive organs and reproductive propagules, which are known as the basidiospores. Earlier in another practical, you have already done the microchemical studies of basidiospores of the Termitophilic species. You may not have completed this, but today you have seen what type of habitat is there. These species which we have brought, you have brought, collected, they are strictly mutualistic, but biochemically saprotrophic. So these are saprotrophs which have lost 130 million years back their independent existence as saprotrophic basidiomycetes. And they are found below Sahara up to Republic of South Africa. They are found in Madagascar, Seychelles, Sri Lanka. These are islands. They are found below Himalayas. They are found from Srinagar to Kanyakumari. They are found from Gujarat to Myanmar and up to Taiwan, Vietnam, Cambodia, Philippines. We have got all the distribution in flora of this. Full global distribution papers we have got, and Dr. Lucy D'Souza has extensively done the literature survey to study the biogeography of these species. We are the first team in the world to study biogeography of, you write, 91 Termitophilic agaricales. As I am holding this species, Termitomyces IV, this is the name of the species, named Dr. Rajar Haim, who wrote the only monograph in French on this species. He is a French mycologist. The entire book is in French language. We use it. Full description, keys are given in there, photographs. Now, what is the importance of genus Termitomyces? Now, this comes under order agaricales in the family Lyophilinaceae. And the importance of this species is extreme range of basidiocarps, ship sizes. That is, the smallest species in this genus, name, write down Termitophilic microcarpus. I will draw it on the blackboard for you to see. For size comparison, in genus Termitophilic microcarpus appears like this. Exactly. Local name in Konkan and Goa is Sutolalani. In Pandan they are known as Singarani. They come in thousands and they are quickly appear. There is no silurica. Except this microcarpus. Size 1 to 2 centimeters only. Cap size 1 to 2 centimeter. Will put it relatively here. Cap size. In microcarpus. The largest of the species in genus Termitophilic in world is not found in India. It is found in Zambia. And because Titan was a famous Greek god, it was named as Termitophilic titanicus. Compare this diameter 1 to 2 centimeters and the diameter of this Termitophilic titanicus is 100 centimeter or 1 meter. Size of a holding umbrella. So one single specimen of Termitophilic hymi of intermediate size, that is around 10 centimeter width. Probably I am telling you without measuring it, and actually it will be around 10 centimeter because 33 years. 33 years. Check whether it is 10 centimeter or around that 11, 9. If 9 to 11 you will be benefit of doubt. If it is 12, you say sir you are wrong. End to end, one diameter. Diameter from margin to margin. What is the diameter? 12.5. So I am wrong by 2.5 centimeters. I will measure again for my satisfaction because you have to draw parallel. You have 12.5. So 25% I am wrong. So that clearly tells you one thing. I am getting over. And wrong. So I am wrong by 25%. I am joking, but the thing is, I told you that 12.5% you place between smallest mushroom microcarpus 1 to 2 centimeter cap diameter, largest is 100 centimeter. You got an idea what I am holding today in my hand, and its weight will be less than 20 grams. What was the weight range of the mushrooms weight?
28.
28, around that, 30 grams. And Titanicus, weight of Titanicus 2 kg, single specimen. One mushroom 2 kilogram. Protein, edible, fungal. So this is the beauty of the diversity of the species distributed in some 40 countries. It is not found in South America, North America, and other areas. Europe it is not found. South China it is found. So today, next part that you are going to learn is how these species are taxonomically characterized, and I quickly go through it. For purpose of taxonomical characterization, most important feature is the habitat which you have seen. The habitat is on ground. Habitat is termite, this one underground fungus comb. It is a mutualistic species. It is a hypogeal state, it is an epithelial state, it is connected by a pseudorhizome which is hollow, tubular, cylindrical, rubbery, okay, and it transits into a solid fibrous type. So for the purpose of description and taxonomical diagnosis, you have to record the field characters where you are collecting the specimens. And why I have kept here 5 specimens, because you can see the size variation within the collection. These 5 specimens represent a collection which is taxonomically acceptable. If you collect only one, very rare cases are there where scientists will find only one plant, or say like Dipcardi goensis, only in one part, Ivona column, like somewhere it is growing, it is not found in our university campus or Saligao, and anywhere. So only there it is found. Only one location. So if you come and tell me, sir, I found a new species, I have to doubtful about it. I say, why you say, sir, it is 12.5 centimeter. But what about this then within the same species? Variation. So how we write the variation? So you measure this, it may be say 5 centimeters. So fruit body diameter is 5 to 12.5 centimeters. These are the largest specimens. So first is always the size of the pileus. So this part is carpophore, fruit, these are the common nomenclature. Fruit body, cap, carpophore, basidiocarp, means this. This is the fertile part. This is simply standing mechanical support, nutrient transportation, no other role. The role of the type is purely to lift the fertile tissue at such a height where winds, rains can carry the spores away from the species. Understood? Otherwise, all plants would have dwarf plants. For purpose of pollination, fertilization, dehiscence, seed dispersal, all fruit-bearing, seed-bearing plants attain height dictated by local ecological conditions, ecophysiological conditions like light and shadow, wind, and evolutionary program. You cannot make a papaya 1 feet tall and yield papayas. The medium, any even hybrid papaya will be definitely a meter tall. Hybrid coconut will not be less than a meter tall. You can't make it a bonsai coconut tree. So what it means? Definitely there is a limit on the type size. It cannot be this size. And this is a reality in genus Termitophilic where the type size is within a particular limit of maximum 10 centimeters. 10 to 12. So type length you describe, type width you describe, width at 3 points. At the base, in the middle, and where it is attached to the cap. Then we come to color of the cap. Color of the cap. Now we use in our lab Color Pro analytical program smartphone based app. Using that app, I just have to take a photograph of the cap and then subject it to color analysis. And all of you have installed it, I believe, ecology or microbiology practicals. Those who don't remember, may please remember when we had done that practical. This is color analysis app. Remember? Who is that? So, use camera. So I am using camera and the storage is मेरा फोटोग्राफर एक्सेप्ट करूँ इसको. Okay. Now this is started. So write down the analysis gives dominant color is dark jungle green. Sorry, dominant color is dark jungle green. It constitutes 39% of the color. Second one is pastel grey, 11%. Then arsenic color, 11%. Platinum color, 10%. Bistre color, B-I-S-T-R-E. Bistre color is this little greyish. This is bistre color, greenish color, greenish. We call it bistre color. Towards brownish color, 9%. Bowl color, bowl, like pale chocolate, 7%. Pastel purple, 7%. Cool grey, 3%. Eggplant, 0.7%. Battleship grey, 0.65%. White, 0.24. Rifle green, 0.22. Black, 0.14. I will not give minor colors. So that color combination defines. So when we have reached from standard printed color chart based, mythological chart based description of different color terms in mycotaxonomy to digital color terminologies and accurate color analysis, which is very important for very precise analysis of species specific characters. So here we use this for style for anything that is color. Understood? So you want to differentiate between style color and cat color, that chart can be published also separately. And for each of these 5 I have to do it, all 5. You may think that sir has taken only color analysis of one, no, all 5. And then analyze all 5 data sets and then write taxonomic description of color terms. Then what makes, that is very important and where your video, microscope and everything becomes useful. This is extremely time consuming and if you ask me, very boring part of the mushroom taxonomy. That is topography.